Hello everyone, so happy you could join me for Chapter 6 lecture, which is DNA and gene expression. These are our learning objectives, although the last two we're going to kind of cover a little bit more in the next chapter. As you can probably guess, DNA is obviously very important, both in knowing who your parents are or what genetic material you get, to diseases, to various genetic conditions, to just what you look like and who you are as a person. One of the things that is incredibly important about DNA is the fact that we can use it to increase the justice system, so make it more precise and more accurate. In this case, this gentleman, Julius Ruffian, was released from prison 2 decades after a wrongful incarceration. He was accused of assaulting a woman, and she thought she recognized him. She misrecognized him, and in the end, it took about 20 years, but he was eventually exonerated due to DNA testing and how far that science has come. So we will be talking about what DNA is and what it does. You've probably seen a lot of DNA in headlines, whether or not you pay attention to it. Everything revolving around stem cells has always been very, very media forward, especially in the last few years, let alone, I think this is probably one of the funniest titles, but relating behavior to various genetic conditions, or blaming your genes because you cheat on your wife or something like that. It's kind of goofy, and really there's not a whole lot to it, but it's still interesting to see that in the media. So the way you can think about DNA is like an instruction manual. If we look at the entire genome, it is quite literally just a huge recipe book for why you look the way you look, why you have the conditions that you have, various things that make you you are in that instruction manual of your genome, and specifically your DNA. These two men, James Watson and Francis Crick, are the ones credited with the exact structure of DNA. We can see their sketch, although they would not have gotten to where they eventually got without the help of, let me find her name, Rosalind Franklin. So Rosalind Franklin really was a scientist who gave her life to the pursuit of knowledge, and she really helped them and probably deserves substantially more credit than she is given. Here we have the structure of DNA. What I want you to kind of pay attention to is, one, it's deoxyribonucleic acid is what DNA stands for, as well as the fact that we are looking at what is essentially a ladder that has been twisted. And the rungs of the ladder are made up of base pairs, and these will always partner up with each other, adenine to thymine and guanine to cytosine. You will never have them without each other. I say that there's a caveat, and that is RNA, but we'll talk about RNA in just a second. So the sides of the ladder are alternating sugar phosphates, so they're just kind of going back and forth between a sugar and a phosphate along those sides, but that's about as much as I want you to know about that. We're going to focus on those base pairs. So genes are sections of DNA that contain instructions for making proteins. That is a very important thing to make a note of. Proteins are kind of the building blocks of everything that makes everything living. We are going to talk about proteins quite a bit. So make sure to underline that, highlight it. We are going to be talking about proteins. Big thing, proteins. These are a couple terms that I would like for you to know. We're kind of going from a more broad look to a more focused look. So initially I want you to know genome, which is an organism's complete set of DNA. From there, from the genome, we are going to look at chromosomes, which is one or more unique pieces of DNA, which for humans we have 46 chromosomes, or 23 pairs, one from mom, one from dad. Next, when you look at a chromosome, if you look at just a specific sequence of DNA on a chromosome, that is a gene. And specifically, if that is a particular location, that is called a locus, which is a position of a gene on a chromosome. Getting smaller yet, we're going to look at alleles. Alleles are different versions of a gene that code for the same feature. The example that I have for you on this slide is these different versions, these different alleles, alleles 1 through 3, that determine daisy petal colors. So that is a trait. That is an example of a trait. So it's any single characteristic or feature of an organism which is affected by those alleles. Not all DNA contain instructions for making proteins. In fact, quite a bit of DNA is going to be something called junk DNA, and that is essentially just DNA that does not code for anything. If we want to get more specific, we're going to call that junk DNA non-coding DNA. When it comes to eukaryotes, they have a large amount of non-coding DNA, which within genes, this non-coding region is called introns, and we'll talk about those more in a little bit. When it comes to prokaryotes, so like bacteria and viruses, they use up most of their DNA. It's mostly centralized to being specific stuff. So about 90% or more of DNA is usable, as opposed to non-coding DNA in eukaryotes. So how do genes work? Here's a little overview. So as the name implies, we have something called a genotype. So this is going to be genes that an organism carries for a particular trait. And remember, that trait is going to be kind of what is displayed. To give that a more specific name, we're going to call that a phenotype. That is the physical manifestation of the genes for a particular trait. So what I want you to get from this is that a genotype is going to affect the phenotype. The phenotype is that physical look of what the genes are coding for. So if we think of the genotype as being like an instruction or a recipe, the phenotype is that end result of how that recipe is going to turn out. Next up, we're going to talk about what is essentially going to be your least favorite thing from this chapter, and I do apologize, it's not the most interesting thing, but it is important that you know it. So from gene to protein, we are going to go from what is inside of the nucleus to into the cytoplasm, which will eventually potentially move out of the cell, who knows, we'll see, through processes called transcription and translation. And this whole process is sort of laid out in this beautiful way on this slide. So I'd maybe kind of pay attention to this for just a second. So our first few steps, we are going to start with DNA inside of the nucleus. How we're going to compare this to something that maybe you can sort of visualize is it's your grandmother, or your mother, your aunt, or your uncle, or I don't know, whoever. It's their cookbook. It is someone else's cookbook. We are going to go through this process of transcription where the gene sequence is copied from DNA to a middleman molecule called mRNA. Remember, I mentioned RNA earlier, and we'll talk about how it's a little different than DNA. So the way mRNA is created is through something called an RNA polymerase, which polymerase is an enzyme. This enzyme is going to unravel our double helix So our DNA strand, which is a double helix, is going to split it right down the middle like a zipper. And from one strand of the DNA the RNA polymerase is going to attach to, it's going to create mRNA, which that mRNA is a single strand. Very important to note. So after the RNA polymerase passes by that one strand or that one section of that gene that we are looking at, our DNA is going to wind back up. So don't worry, it doesn't stay unwound. It will wind back up. This is like copying the recipe from our cookbook that we're looking at. We are writing it down somewhere else, okay? So we are making a copy. Very important thing. From there, our mRNA is going to kind of pinch off and then exit the nucleus through the nuclear pores and end up being out in the cytoplasm of the cell. Here, our mRNA is going to go through something called translation. Translation, as the name implies, is we are going to translate that gene information into a protein molecule. So that is what we are going to do. So with that process, we are going to use ribosomes. Remember, ribosomes have everything to do with proteins. This is going to be like combining and baking our ingredients. And then that protein molecule that it ends up creating is going to be like the finished product. Maybe it's chocolate chip cookies or whatever. So let's talk about this a little bit more specifically with very specific steps for both transcription and translation. So in transcription, the information coded in DNA is going to be copied into mRNA. What is going to be changed between DNA and mRNA is going to be that we lose thymine, so that little T base pair. And instead of a T, our adenine, our A, is going to pair up with something called uracil, which is going to be written out as just a U. So we go from DNA to RNA by dropping the thymine and instead using uracil, a U. So big difference. You always know you're looking at DNA if it has a T. You know you're looking at RNA if instead of a T, it's a U. And you'll see what I mean here in just a bit. So let's go through a few of these steps when it comes to transcription. So the first step is going to recognize and bind. This is going to be once the RNA polymerase, remember that enzyme recognizes a promoter site. And a promoter site is just going to be essentially where a sequence in a gene is indicated that there is a starting point. So that is just our starting point, if you want to write that down. And that is where the DNA is going to begin reading the gene's message or instructions or recipe, however you want to think of it. So there is a starting point. There is also an ending point, and we'll get to that. So from there, our RNA polymerase is going to work down one side of our DNA, and it is going to start transcribing. As the DNA strand is processed through the RNA polymerase, our enzyme, the RNA polymerase is going to build a single stranded RNA copy of the gene. So that is the mRNA transcript. So that is just our strand of mRNA. Remember, instead of a thymine, we have uracil. The next step is going to be to terminate. That is where we have our ending point, our stopping point, and that is when the RNA polymerase knows that it needs to detach, and that is the end of the gene that it is transcribing. From there, the last little step is to cap and process. This is sort of like a final editing portion of the mRNA where you put a front cover and a back cover to this RNA, as well as you are going to cut out those introns. Remember, the introns are those areas of non-coding DNA. So we don't care about it. It's like excess writing. We don't need it. It's pointless steps. we can just get rid of it. And that is transcription. Remember, transcription is just copying. In translation, the mRNA copy that we just made of the information from DNA is used to build a functional molecule. And in this instance, we are going to use something called a transfer RNA or tRNA, which is going to be molecules that translate the mRNA code by linking specific bases on the mRNA with a specific amino acid that will be used to build a protein. And you'll kind of see what I'm saying here in just a second. So what you can think of this tRNA as is sort of like a price checker, or like a cashier's point of sale machine. So if you run the product by that machine, it's going to read it and then put it up onto the receipt, right? That is essentially what this is doing, but instead of building a receipt, it's building a molecule, which is going to be a protein using amino acids. So we have that attachment site where it is reading the parts of the mRNA 3 at a time, 3 base pairs at a time, which is called a codon. So as it reads those 3, it creates something called an amino acid. Amino acids are going to be 3 base pairs long, and you can read a base pair by using the codon table. So this is kind of the key to the codons, if you will. So the first part is going to be that first base pair, which is going to be a U, C, A, or G. The second position is right along the top. Again, U, C, A, or G, and if you can't find it by kind of hunting in that general area, you can always look on that third position if you need to. So for example, if your codon translates for something, a type of amino acid with the letters A, U, G. So that is what we get from our translation. Okay, so we went from DNA to RNA, and then it is being translated. Okay, so that is the other side of the mRNA with a U. All right, so we get something with AUG. We're going to go down the side of the first position to A, go across the top to the U area, and that's going to bring us to that first row and that first column and 3 rows down. That's going to bring us to that box. If you can find AUG, which you may have already found it, you know that it is something called methionine. Methionine is a very special little amino acid because it is always the start of an amino acid chain or a protein. Okay, the ending codons are going to be UAA, UAG, and UGA. Those are always a stopping codon. Okay, let's talk about translation. First, we are going to recognize and initiate the protein building. So we are going to be at the start sequence, which remember, that's always methionine. Methionine will always be our starting sequence, which is AUG. It is going to be recognized by a corresponding tRNA. Remember, that's our little price checker molecule, and 2 ribosomal subunits. Those ribosomal subunits are going to be important because that's going to be like the factory that our tRNA is going to enter into and thus be able to work through. So those ribosomes are sort of like a factory, the tRNA are sort of like the workers, and then our amino acid protein chain is going to be the product that we are creating. The attachment site of the tRNA molecule is going to bind to the mRNA as the ribosomal subunits assemble around them, again like a factory. So that mRNA is going to be moving through our ribosomal subunit where those tRNAs are going to come in and match up to those 3 base pairs at a time until it finishes, right? So I'm getting a little ahead of myself. So the second step is elongate. As the ribosome moves along the strand of mRNA, each new amino acid carrying tRNA molecule is going to bind to the next 3 bases on the mRNA. After the ribosome attaches the growing protein chain to the new amino acid, the tRNA molecule detaches from the mRNA and floats away because we don't need it anymore. It has already done its process. The last step is to terminate. So once the ribosome encounters the 3 base pair stop sequence, which we talked about that on one of the last slides, the protein assembly is going to be complete and translation will end. From there, both the protein and the mRNA molecule are released from the ribosomes, and sometimes the mRNA can be used again, or it can be used as a kind of a base for another piece of mRNA. It could just kind of go float out in the cytoplasm. It might leave the cell, but who knows? It's going to go live its life. And then that protein is then going to be either used in the cell, or it's going to leave the cell and be used outside of the cell. So here we have some questions, and you can pause this at any time to test your knowledge. All right, from there, we're going to talk about genes being regulated in several ways. So we've talked about genes and how they kind of make you. So here is one of the ways that we are going to regulate our genes, and this doesn't mean necessarily maintenance. So kind of throw that term out of your head as far as synonymous with regulation. We're not talking about maintenance. So gene expression is the production of the protein that the gene sequence codes for. Another term that we need to know that goes hand in hand with gene expression is gene regulation. This is whether a gene is turned on or turned off. So this is going to determine essentially what proteins are being produced or not. And this can be a good or a bad thing. If certain proteins are, certain genes are turned off and certain proteins are not being made, this could end up turning into lactose intolerance. Or it could be, if the gene is turned on, it means you have blue eyes. You know, it just sort of depends on whether or not a gene is turned on, and that is how we are talking about this regulation. Your environment is often going to have regulatory effects on your gene expression as well. So it can be diet, it can be exposure to various things. It just depends, but that does have a lot to do in your gene regulation. One of the things you need to know is that cells have a wide variety of mechanisms for controlling gene expression. One of those is transcription factors. These are going to be proteins that bind to specific regulatory sites on the DNA. You have a positive control and a negative control. Respectively, a positive control initiates or speeds up gene expression, while contrary to that, negative control is going to slow down or block gene expression, which is then going to hinder or exacerbate our protein creating abilities. So gene regulation is extremely important, especially during your initial development as an organism. So this is going to be like in utero. Very specifically, it's going to be very early on in utero. So pretty soon after conception, essentially, when a sperm cell meets an egg cell and creates a zygote. From there, you're going to have a lot of cell division going on, and specifically, this is going to determine, you know, your body composition, the development of your brain, the development of your bones, the development of muscle instead of, you know, maybe skin tissue. You know, it's very specific, and, you know, to say the stars have to be aligned is sort of an understatement. There's a very complex process that goes into making sure that these genes are going to regulate and express properly in order to create a normal functioning creature. And that doesn't just mean humans. That can be plants, that can be animals. It can be literally anything that is considered a living organism. It's very important. So what causes a mutation and what are its effects? Remember on that last slide that I said it's very important for those things to happen in a particular way, and especially that it happens in general. Well, in that process, we can have problems. What these problems are at the kind of smallest level. So if there's an issue in our DNA sequence, an alteration, if you will, that is considered a mutation. Mutations can be detrimental, or rarely, but not rare enough, I suppose, can be beneficial. When it comes to mutations, mutations are essential to evolution. It's very important that mutations occur, and you might be thinking, but mutations sound like a bad thing. They're not always. Sometimes they can be really bad, but sometimes they're neutral or good, and they can affect the ability for an organism to reproduce better than, say, another individual of the same population. Pretty much everything that we see today has been caused by some type of mutation to get us where we are now. And just to make sure we know, some mutations can be silent, or, you know, basically not affect you in any capacity. Some mutations can be good. An example of a mutation that is, it's not the best one, it's pretty, but blue eyes. Blue eyes in humans is a mutation. So any color other than brown is technically a mutation. And while, you know, blue eyes or green eyes or violet eyes, they're very desired, you know, they're beautiful, absolutely stunning. They do make it to where those individuals actually have a harder time seeing than people with darker eyes or brown eyes. So, fun stuff. So we have these types of mutations. One of the most kind of can be detrimental is mutations that occur in sex cells. So sex cells are X or Y chromosomes. Okay, what happens with these cells is that males typically get the short end of the stick, especially when it comes to humans. Male pattern baldness, as well as color blindness, is actually caused by mutations and sort of bad genetics, if you will, that are passed on from mother to son. And we'll talk about this more when we talk about various types of cells, but essentially the X chromosome is a lot bigger than the Y chromosome, about double its size. So there's a lot more information that gets passed on in that X chromosome versus the Y chromosome that is given from the father. So what happens with that is males end up getting whatever condition it is that's on the X chromosome, because there's nothing to combat it with the Y chromosome. Again, we'll talk about this more later in the semester, but that's just a little bit of stuff pre-lecturing from that section that we'll talk about soon. And then there are two major types of changes to DNA that can occur: point mutations and chromosomal aberrations. With point mutations, there's nucleotide substitution, where we are substituting that single nucleotide we have a nucleotide insertion, where we are inserting some nucleotide into a spot, and then nucleotide deletion, where you are removing that nucleotide from that spot. So insertion and deletions can be much more harmful than substitutions because they can alter the reading frame for the rest of the gene. So if you put something in, it's going to move everybody else down. Or if you take something out, it's going to move everybody else forward. So all those nucleotides are going to be moved around, as opposed to a substitution, which is not going to change further down the line, change the nucleotides further down the line. So those are point mutations. I would make a note that with point mutations, it's a single nucleotide that's changing, as opposed to a chromosomal aberration where you have full sections that are being altered. So we have gene deletion, gene relocation, goodness, try saying that 5 times fast, where you are moving or relocating to a different chromosome entirely, and then gene duplication, which, you know, obviously you're just making a copy, or there is another copy further down. So we do have some chief causes of mutations. Sometimes we have spontaneous mutations that arise by accident during DNA replication, as this is sort of like a weird pseudoscience phrase, but you probably heard it. Every 7 years, your whole body just regenerates with new cells. There's a totally new you every 7 years, which is kind of a weird misinterpretation of how cell division and DNA replication work, but regardless, about every 7 years, I guess, yeah, your cells have all completely changed themselves out from what they originally were, but I mean, it's a weird phrase. Regardless, back on point. So as your cells regenerate and create new cells, so if you think of your skin cells are constantly being pushed up, sort of like a push pop from a base layer, working their way up, so that the top cells, once they get to the surface of your skin, they slough off, and that is what is essentially dust everywhere in your room or wherever is mostly skin cells, which now you're going to go home and absolutely dust everything. But as that occurs, we are having cell division occurring relatively rapidly, not crazy fast, maybe not as fast as you think it does, but pretty quick. And while that is occurring, we are having DNA replication occurring. So this is going to be a process of mitosis, and we'll talk about mitosis later, but that is cell division. And as that occurs, we have to replicate our DNA for those new cells to exist. Because if you remember cell theory, one cell gives rise to more cells. You have to have a starting cell in order to get a new cell. So DNA replication has to occur for that parent cell to give that DNA to its daughter cells. From that process, you may get accidents of mutations, which will essentially cause issues potentially like cancer. Environmental factors that can cause mutations are radiation-induced mutations and chemical-induced mutations. Chemical-induced mutations include smoking. I don't want to get on my soapbox again, but I'm gonna. Smoking is probably one of the worst things you can do to your body, and that includes vaping. So if you are inhaling anything other than just air, you are going to cause problems for your body. And I know that there's a lot of like safer ways to smoke, but you're still taking something in to your lungs and exposing your very, sensitive lung tissue, as well as the tissues of your throat, the tissues in your sinuses, in your face, as well as, you can't help it, you're going to be ingesting some of that too into your stomach. And everything you do that you take into your body like that is not meant to be there, such as, food or water or air, can cause mutations or problems with the lining of those areas. So those cells that are exposed to what you are ingesting or inhaling. So try not to do it if you can, help it. Another thing that people might not think about as being a way that you are increasing your risk of getting a mutation is tanning. Tanning is honestly one of the worst things you can do to your skin. It might look really nice, but you are increasing your chances of getting skin cancer, especially if you burn like the young lady here in the middle frame. That is something she would maybe want to keep an eye on, especially if she's doing it often. Okay, moving into the last little bit, we've got these faulty genes coding for faulty enzymes that can lead to sickness. I did talk about how genes, if they don't code correctly, can cause issues. In this instance, I believe this is talking about how your ability to kind of flush out chemicals is affected by this. Another example is lactose intolerance. If you have faulty genes that code for faulty enzymes, it could inhibit your ability to digest certain things. This includes like flour, so gluten, and lactose is the other one that I can think of right off the top of my head that's pretty popular right now. So if you have these faulty genes, you're not going to be able to produce enzymes that help you digest those various things. You just can't digest them, and so it makes you sick if you consume it. So drinking milk, if you're lactose intolerant, can cause a lot of digestive discomfort and, illness. And that is the end of chapter 6. This was actually a lot shorter of a lecture than I expected, but I hope you enjoyed it, and have a wonderful day. Don't forget, make good choices.